now. So take it away.
Then I'm going to go ahead and get started. And thank you for joining us today for the Kentucky Inbree seminar series. I will be introducing the UofL Genomics facility, and after that, Julia Aebersold will talk about the electron microscopy core. So our UofL Genomics, hold on, you can see the slide. Yeah, okay. Our UofL Genomics facility is located in the CTRB building on the Health Science campus, and our team is Dr. Wolfgang Zacharias, the director. And then there is me, I'm Sabine Waigel, the assistant director of the core. There is Shirong Chong, our research scientist, Venila Arumugam, she's our senior research tech, and she has been with us for over 15 years now. So she's got plenty of experience in different library preps. And recently, Shiki joined our lab. She has a master's in bioinformatics and is now working on her PhD in molecular biology. So she's going to be a great asset to our team. Just making sure you can all see my slides okay, right? Yeah, all right. So with regard to the services we provide, we try to provide a full life cycle support for your experiment. So upfront, we can provide consultation, help you plan your experimental design. We can provide cost estimates. We also provide assistance for grant proposals, support letters, manuscript paragraphs for method sections, etc. With regard to the lab services, we provide quality and quantity assessment of RNA and DNA. We also prepare Illumina next-gen-seq libraries. We perform the sequencing runs for those, and more recently, we have added single-cell sequencing to our repertoire, and I will talk more about that towards the end, but it does give us a gene expression level on individual cells in a mixed cell population. And then the data that we provide goes to the Kentucky Inbree bioinformatics core that runs the analysis. But after that, we can still help with data interpretation and validation. We have a real-time PCR for individual gene validations, and for data interpretation, we have access to MetaCore. And that is a software that allows you to see which functions and pathways are overrepresented in the genes that have changed in your experiment. We also offer training. That can be training in wet lab procedures, but we can also organize seminars and workshops on technology sessions, seminar, software training, etc. Let's look at the equipment we have. We have 2 Illumina instruments. We have a MiSeq and a NextSeq 500. They are suited for 2 different applications. The MiSeq has a low throughput, but a little longer read length. It can do 2 by 300 base pairs. And the NextSeq has a higher throughput. It gives us more reads, but the reads are shorter. So we use the MiSeq for amplicon seqs, like 16S, when we need a read that is 600 bases long. But we use the NextSeq for counting applications like RNA-seq. We also have a 10x Chromium controller, that is our single cell platform. And we have an ABI real-time PCR instrument. In addition, we have several other small equipment available. There is the bioanalyzer, the nanodrop, and the qubit. Those 3 we use to assess the quality of your RNA or DNA. And they all tell us different things. For example, the Agilent bioanalyzer is electrophoresis-based. So there we can see the size distribution of your RNA fragments. So that will tell us how intact your RNA is. The nanodrop, on the other hand, is a spectrophotometer. So there we can see if there are certain contaminants in your sample, if they absorb similar to that spectrum. Like we can see if there are phenol contaminations from the isolation. We can see if there are salt carryovers from column kits. And those might interfere with downstream processes. The qubit, on the other hand, is a fluorometer. And that is the most sensitive quantification method we have. While, let's say, the nanodrop can't really distinguish RNA and DNA, they both absorb at 260 nanometer. The qubit has specific dyes that can distinguish RNA from DNA. It can even distinguish double-stranded DNA from single-stranded DNA. So we run sample QC on all three instruments, and they all tell us something different. So in addition, we have a countess for cell counting and a GentleMax octodissociator to dissociate tissue very gently to keep your cells alive and well for single cell sequencing. We also have a Covaris S220 that is a fancy sonicator. It's a $40,000 sonicator that can break your DNA and chromatin very accurately. So you can program it to get quite accurately the size that you would like. We have had researchers come on campus from the regionals to use these instruments for the day. So they are open and accessible for researchers and our regional institutions. There is now also, we have long read capabilities on campus now as well, and they are also available to Kentucky Inbree regional institutions at internal UofL pricing, just like the Illumina sequencing. So there's now a PacBio Sequel 2 and the Oxford Nanopore technology. And these services are available through the UofL Sequencing Technology Center, and again at internal rates for the PUIs. Let me explain quickly what the difference is between short read platforms and long read platforms. And for that, let me turn on my laser pointer here. So ideally, at least from the viewpoint of the biologist, we would be up here. We would get data that has long reads and a whole lot of them. But right now, there is no platform that can do that. So everybody's aware of that. So PacBio is trying to move there, Illumina is trying to move there. But right now, we have to make a choice. Do we want many reads or do we want them to be long? And the answer to that will depend on your application. We need many reads for counting applications because so that is RNA-seq, microRNA-seq, ChIP-seq, RIP-seq. The reason for that is if you want to try to pick up lower expressed genes, you just need a high sampling rate. On the other hand, you need long reads when you're trying to assemble a genome. That's the only way that you can sequence through repeat regions and know where that goes in your genome. Also for splice variants. If you want to get a single contiguous read of a gene, you will need a long read. So it will depend on the questions you're asking and the research focus, which platform is most suitable. And we are there to guide you. So if there are questions, just reach out to us and we will guide you to the proper platform. And hopefully in the future, we will be somewhere up here, although bioinformatics might have more hesitations there. So with regard to the next generation sequencing services we offer in the genomics core, we mostly do gene expression, which includes mRNA-seq. So for that, we enrich the polyadenylated RNA molecules in your sample. We can also do a total RNA-seq, for which we would deplete ribosomal RNA, microRNA-seq, commonly performed. We can also study RNA, DNA, protein interactions, such as ChIP-seq, RIP-seq, etc. We run a good amount of 16S metagenomic experiments, as well as other amplicon-seq that's very similar. And we can perform small genome sequencing. So we don't really have the throughput to do mammalian genomes. That would just not be cost effective on these platforms, but we can sequence bacterial and viral genomes, and boy, have they been in demand lately. And our newest technology is the single cell sequencing with 10x Genomics. And one of my last slides, I will explain more what that does. The pictures here on the right don't belong to any specific technology. It's just to show, again, and a shout out to the bioinformatics team that can create really beautiful images for your manuscripts. So we have been quite busy, at least over the last year, year and a half, 2 years now, with SARS-CoV-2 sequencing. Prior to March 2020, when you were dropping off RNA samples for an RNA-seq experiment, we would have told you we need intact RNA. Ideally, they would have a RIN number of 10. What that means is that we can see clear ribosomal bands and very little degradation. We would also have told you that the total RNA amount we need is about a microgram. But then March 24th, 2020 comes around, and a researcher walks into our lab with these 3 samples. These were RNA isolations from nasal swabs from 3 COVID-19 patients. We had RINs of 2 point something. You see there's barely anything left of the ribosomal RNA. Instead, everything is degraded. Down here, we have small fragments. More than half of the sample has a fragment size of less than 200. So we went out, worked with manufacturers, found a kit that is able to preserve these small fragments in our sample prep, and then even worked with Illumina to modify the sequencing parameters on their instruments to allow for that kit. And with that, we were able to actually assemble full genomes out of these very degraded samples. And we were then able to map these genomes phylogenetically to the other genome sequences we had available at that time. So this was end of March 2020, and these were all the genomes that had been sequenced and deposited. And there are our 3 Kentucky samples. Emboldened by that, the Envirome Institute at the University of Louisville reached out to us and said, well, do you think you can do a whole genome sequencing from wastewater? So we said, well, why don't we give it a try? And we found a kit that used an amplicon panel that was tiled across the whole genome. And we were able to, so the Envirome Institute is doing the sampling. They sample 16 locations in Jefferson County and extract the RNA from those. And these 16 sites include manholes and wastewater treatment plants. And we then get the RNA, isolated RNA, create cDNA, and then sequence that, and are able to assemble a pretty decent covered whole genome of SARS-CoV-2. The yellow region here is the spike region. That is always of specific interest. And every week, again, a shout out to our bioinformatics team. Our bioinformatics team can put together these beautiful reports where our city can see which variant can be found at which location in the city, and even provide a map. So the city has some guidance as what we can currently see in the wastewater. And the reason why I am actually bringing this up is not because I'm telling you that you can bring us degraded samples now, and that is perfectly fine. The sample quality is still very important. The better your sample quality is, the better your data will be, and that has not changed and will not change. But I wanted to point out that we're pushing boundaries. So if you have been told something is not possible a few years ago, go back and ask again. Another area where we are pushing boundaries is the single cell sequencing. And I want to go over that in a little bit more detail and explain what that is, because there are sometimes misunderstandings. Some people think single cell sequencing is actually the sequencing of a single cell, but it is the sequencing of an individual cell, but up to 10,000 of them in your sample. So the way that that works is, in the instrument, you feed your cells in one channel, and in another channel you have, they call those barcoded gel beads, and they are fed into an oil and form an emulsion. So then you have a little reaction vessel. That is your little water aqueous bubble in your oil phase. And ideally, each one of these bubbles will contain one cell and one gel bead. And that gel bead has a barcode. So I've always wondered, how do reaction work on gel beads? But it is actually just the gel to deliver the reagents, and then this emulsion is heated and the gel melts. And then you have like a micro vessel of reagents and your RNA in that one bubble. So these barcodes label your RNA from that cell, and then the emulsion is broken, and you treat your RNA like you do any other RNA-seq, but now you have a marker which cell that RNA molecule, that RNA came from. So the data you get out of these experiments looks a little bit different, because now you don't have a bulk RNA single table with results. Now you get a transcriptome profile for every cell in your sample. So you get a cluster similar to this, and each dot on this plot represents a single cell. And you can then, so you can see that there were different cell types in this sample, and then you can try and figure out which cell type is which cluster. For that, you know which genes were overrepresented in that cluster. And as that is often not enough to identify a cell type, we can also tag surface markers. So that is where we're getting more into the multi-omics area. We can actually label your cells with antibodies before sequencing, and then we can not only see the expressed genes, but we can also see which surface markers. The antibodies have oligotags, so we sequence those as well, and we can see which surface marker is present in that cluster. In addition, it's possible to do immune profiling on these cells. We can do T cell receptor sequences. We can do B cell receptor sequences. We can also do a tag-seq, see open chromatin structures. So this is enabling more and more multi-omics approaches on a single cell level. So as if this is not exciting enough, the next step is going to be to do the whole thing in tissue. So this is called spatial transcriptomics. We have not incorporated that, but are looking at that later this year. So for spatial transcriptomics, you don't have your cells in suspension, but you take a tissue slice, just like you do for your histology experiments. And you deposit that tissue slice on a slide. But this slide has markers that mark the XY coordinates on the slide. So now you place your tissue on the slide, you permeabilize the tissue, and your RNA diffuses down onto the slide and gets labeled with the XY coordinate of where it came from. Again, you take all of the RNA, analyze it as we did before, but now we have an XY coordinate label for each RNA molecule, and we can map it back to the original histology image. So we can see which gene was expressed where in that tissue. And for analysis, it can go then two different ways. You can take the histology image and say, these are regions of interest, and I want to compare these. So I can compare the red region here to the gray overlaid region and see which genes are different here. Or you can have the analysis actually driven by the clustering itself. You can let the data drive it and tell you where are different groups of cells, and then see what is actually the difference in those. What I think is important to point out is that for spatial transcriptomics, as well as for the single cell sequencing, you can see the data analysis is becoming much more interactive, or it has to be more interactive for the researcher. It is no longer a single spreadsheet you get with genes changed in your comparison A versus B. You will actually have to interact with your data, figure out here which clusters are the clusters that I'm interested in, or here which regions of the slide are actually my regions of interest. So it will become more and more interactive for the biologists to learn these software tools to be able to look at the data. One other thing we're trying to increase is the outreach to Kentucky Inbree regional institutions and education. So last year we had a summer genomics workshop for the first time. We invited 4 students to join us on campus for a week. And we spent the week just running RNA QCs, prepared an RNA-seq library. We sequenced the library, checked the QC of these sequences, and then even talked a little bit about the data analysis that was going to come after that. I've been told it was one of the most fun activities we had on campus that summer. And we definitely want to do that again. So if you know, if you have a student that you think would benefit from a workshop like that, please let me know. The same is true for any other ideas or suggestions you might have of how we could better support your research. Yeah, I've put my email in the chat. So if you could please, yeah, just reach out to me and let me know how we can better support you, and we'll be happy to see if that can be incorporated. That's all I have. We actually still have a little bit of time. Are there any questions?
I thought that was great.
That was great. Can I, somebody here, let somebody else. I have one question. Jason, did you have a question?
Yeah, the summer workshop, is that for undergraduate students or graduate students or both?
It is open. Yeah, it is open.
All right, so it's a kind of a application or merit-based or need-based?
We will figure that out for next year. Right now, we're a little short on time. So I think it'll just be a first come, first serve, because there will not be much time to
So not this summer, but next summer.
Right. No, I'm trying to still see if we can pull something together for this summer as well.
Okay, great. I'll be the first one to send my students.
Okay.
Yeah, thanks.
Sure.
Great. I was curious about the spatial transcriptomics.
Yeah.
So when you cut the slices, so I guess they have to be fresh or frozen, or what?
I think they can be frozen. Yeah.
Frozen, but not fixed, of course. But yeah, they could be frozen.
We have a solution for fixed tissue now as well.
Really?
Yeah.
That's interesting. Then do they have to be a certain thickness to make that work? I mean,
yes, I
guess if you had it really thick, you'd just be pulling the cells from the side next to them.
And also you need to be aware of the diffusion. You know, you have a certain thickness to diffuse to the bottom, and that is about determining your size of spot you can get, because you will get also diffusion to the side. Or yeah. And
then do they have different like, can you get different spatial resolution? Like, I guess if you have they'd have the dots would be far apart, but I would like to put a teeny little piece and have all my dots.
Yeah, right now it was still at 50 micron. So not quite single cell, but that is increasing. So that seems to be getting better quickly.
Oh, okay. That's cool. Okay, thank you.
Yeah. And for the FFPE, they have just a probe-based chemistry where they can actually pull out then RNA fragments, even if it doesn't have a poly-A tail. So it's very promising.
It would be neat to do. It would be neat to possibly do a TEM image also, if we could overlay it somehow. That would be kind of neat. Anyway, we could discuss that later. But what was the thickness again that it has to be for the spatial transcriptomics?
I would need to look that up. Yeah, I am not sure, but I would guess as close to your cell level.
Okay.
Yeah.
Well, we could help out with that. I mean, yeah, if you're No, that's probably in the micron. range. You're
Probably a
thinking?
cryostat is the best option. for
Yeah,
Getting
yeah,
something, but
yeah, probably 3 to 5 microns, you think? Around there?
Probably a little bit more. I can check.
Yeah, okay. I mean, sorry, I'm digressing. I think your workshop is cool. If I was a student, I would definitely go.
Yeah, I
for
know
it.
the microscopy.
So Sabine, I have
Well,
two questions, follow up on
sure.
what Martha and first
Okay.
question I asked. So with the workshop, have you posted anything about that? I mean, is there like, okay,
No,
do you know like when it will be in the summer?
I
or-
was thinking of early June. I
Okay.
just got off the phone with Susan trying to see if I can arrange for housing on such
Yeah.
short notice. And also some lab training. There are always these other issues to consider, but I think we can make it work.
Okay.
Yeah.
Yeah, if you get something together, that would be good to like send out to the
Yeah,
leads.
I
and
put it in
can-
the newsletter and I will send it out to all the leads.
Okay, I'm
But
sorry, is
again,
it in the
if you
newsletter?
Reach out to me also, then I can make sure you get it.
So it's in the newsletter?
I will have to put it
Okay.
in the newsletter. Yeah.
Okay, and then with the spatial transcriptomics, can you put multiple sections on a slide, or is it just kind of one section?
Theoretically, you could do that, but it's a rather small area. So then it becomes more a question if you can actually manipulate the tissue.
Okay.
Yeah.
I'm trying to get my head around it. It sounds really
Right,
cool,
right,
but
right.
I'm, yeah,
Yeah.
okay,
And then there's always the cost of the sequencing, and the more area you have to sequence, the higher the sequencing cost.
okay. It sounds really, so do you have it up and running yet?
No, not the spatial.
Do you have any idea when you might? Sorry to push you on this.
No, it's a lot of excitement about it. Yes, we were
Yeah.
looking at it later this year,
Okay,
maybe Q3.
Cool. Any other questions before we move on to Julia? Okay, I guess you can share your slides.
Sure. Martha, I had a quick question. Is this
Mhm.
is this mostly U of L folks today?
We have Moorhead, UofL, Murray, and KU.
Oh, okay.
So,
Awesome.
yeah.
Well, that's fantastic.
Yeah.
Okay. All right. Let me do the share thing. Share that. Yeah. Do the share thing. All right. And can you see my slides?
I don't have it yet, but it's coming. Yeah. Okay.
Okay.
Right.
It says participants can now see your screen. Is that right?
Yeah, it's not in presentation mode yet, but
Yeah. There we go. Okay. How are we now? Are we good now?
good, good.
All right, fantastic. Okay, so anyway, I'm really excited to talk with other researchers beyond U of L and UK. I mean, of course, we like UK, but it's always nice to talk with Northern and Moorhead and Murray and so forth. So, by the way, Moorhead has an awesome space program at their facility. Huge, nice, big satellite dish up on one of their hills. Anyway, again, Aebersold. And I actually have a variety of hats on, but within Kentucky Inbree, we have a host of different cores and so forth. And specifically within Kentucky Inbree, we have the electron microscopy core as well, and that is actually comprised of different capabilities across the state, specifying in electron microscopy. Okay. That is going to be, of course, here at U of L, at UK, and then also at Western Kentucky. Specifically at U of L, it's in the Micro and Nanotechnology Center, which is the other hat that I wear. I manage that core facility. A core facility is like a shared facility where it's open to researchers and external people. You come and use the tools, and then you get charged fees for that. So it's not like a private lab. And in all three cases, that they kind of operate in a similar fashion. Down the road from us is the EMC at the University of Kentucky, which is the Electron Microscopy Center. And then down at Western is more geared towards education and training as opposed to research, but they do have capabilities down there at the Ogden College of Science and Engineering. So what is the purpose of the Kentucky Inbree Electron Microscopy Network? Okay, that's a mouthful. So that's basically to share all of these capabilities, not to, to these 3 universities, but definitely to reach out to the other institutions within the state, to where we can say, hey, we're here, work with us, we'll help you out, we will perform services for you, or you can actually come to our facilities, get trained. We definitely want to share these capabilities because they are in shared facilities and are meant to be used by the researchers of the state and so forth. Okay, so what I'm going to focus now on are, we have other capabilities within each of these cores, but I'm going to focus specifically on TEM and also SEM imaging. Now, if you've already done a lot of this and understand this part might be, kind of boring and you may already understand this, but there's actually a lot of researchers that they're very familiar with their certain wheelhouse of skill, of techniques and data analysis that they use. And they may not be aware of what electron microscopy can offer and so forth. So I'm going to actually kind of offer a little bit of an explanation. It's not like, you're going to get quizzed at the very end or anything like that, but we do find that there is a need to explain what the 2 different types of in this case, 2 different types of electron microscopy that we offer within the state. And the first one is TEM imaging, and the other one is SEM imaging. And primarily the way that I emphasize the difference between the 2 is that with TEM imaging, you're actually looking through samples with electrons, okay, as opposed to like an optical microscope, with this light and so forth. But in this case, you actually have a sample. Let me just go jump to this next slide here. You have an electron beam that's up here at the very top. It's going through several lenses and condensers and so forth. You actually have your sample right here in the middle of the beam that is sitting on a very special grid. It goes through another lens, and then the image, the electron beams, the electrons, that get through the sample actually end up on the detector here and form your image right here. So I like to emphasize TEM is through the samples with electrons. So here is a sampling of the TEMs that are offered at each of the different schools. Here at UofL, we have the Hitachi 7700. Down at UK, they have the FEI Talos F200X. And down at WKU, they have the GEM 1400 Plus. I will also say that I'm not sure about WKU, but both the UofL and UK instruments are under service contract, which is one of the benefits of having these in a shared facility. So to get these really thin sections, and this is also part of the reason, Sabine, why I was asking what your thicknesses were and so forth, to be able to send electrons through these samples and so forth, they have to be extremely thin, roughly about 80 nanometers. So what we do in the preparation scheme and so forth is we have this instrument right here. It's called an ultramicrotome, and we section these down to about 80 nanometers. Around there. You can be a plus or minus from there, but they're literally so thin that they will float on top of water. And then we put them onto these special grids right here, which are perforated or in the form of a slot right here, where, and then there's like in this particular case on the slot grid, there's another very thin membrane called FormVar that sits on top of that. And then you put your actual sample on top of that, and then the beam shoots through both of these grids, and then you get your image from there. So I'm not going to go through all of this. This is up here mainly for to give you an idea of what's included. But there are basically 3 different kinds of samples that we can work with. Okay. Number 1, we can definitely work with cultured cells. Usually this is a case where you're culturing, you release it, you centrifuge it to form a pellet. We then fix it, and then we go through our whole process right here. So I'm not going to go through all of these steps, but essentially what you're doing is a process of fixing, then staining, washing, fixing it in, or making it into a permanent structure of a resin material. You then slice that, and then you stain it again, and then it's finally ready for imaging in the TEM. That roughly takes, if you're going full tilt and you have no interruptions and no email, which is not an ideal world, you could probably do this in about 4 days. but with all the interruptions, it takes roughly about a week to do this from here. So cultured cells is one. Tissue is definitely another area that we can do this as well. And then finally, the last one is formalin fixed, or the FFPE samples as well. We have not done with this one right here, but we know that there are loads and loads of samples out there that are FFPE, and we want to try this. The resolution may not be as good as with tissue or cultured cells and so forth, because we are kind of relying upon how the cells were initially prepared and so forth. But we definitely want to give it a try. One thing that is hypercritical with sample prep, or excuse me, with imaging, is always sample prep. If you have, it's kind of like garbage in equals garbage out. If you have bad sample prep, obviously you're going to have bad imaging. And what's even more critical is the finer and finer resolution you want to go with, sample prep becomes even more critical. If you're wanting to look at stuff that's like 100 microns, 50 microns in size, you can kind of get away with things and so forth. But if you're wanting to start to look in the nanometer range, like 200, 600 nanometer range, then sample prep and adequately fixing the materials and so forth is extremely important. Okay, so enough of that. Here are some of the stains what we use. These are not human friendly, so we do have to use these. We have to be very careful. And then from there, we can form our TEM images. And these are some of these right here. These are macrophages that were that the researcher was specifically interested in looking at the mitochondria structures that we have here. And then I'm going to move forward with SEM imaging. Okay, so SEM is kind of like the sister to TEM, where again, we have 3 different types of instruments at each of the facilities. And I will say UK actually has a few more SEMs as well. They also have FIBs and a few other instruments as well. And if you're interested in those, I can let you know offline. The instrument that we have over here at UofL is very nice. It's only about 2 years old, and it also has the nice capability of gathering really nice images of biological specimens. Okay, so typically SEM work is where you can put in non-organic metallic type images and so forth, and you get stunning images. Well, biological specimens aren't like that at all. They're ooey gooey. They like to charge. They don't conduct electricity very well. So typically you have to get an instrument that can run with these types of samples and run at different vacuum levels as well. So what is an SEM? So the primary difference between SEM and TEM is that you're looking at the surface of your structures as opposed to looking through with like TEM. So in the example that I've got here, you've got a spider. This is actually the eyeball of the spider right here. Kind of looks like a fly. And then over here on the right side, we actually have cultured glioblastoma cells or brain cancer. Now the scale bars on these are pretty large, okay, but don't let that fool you because this instrument can read down into the nanometer resolution. One thing I will say though is that when you're looking at biological specimens in the SEM, it becomes harder and harder to look at stuff in the nanometer or the smaller scale bars. You can get great images with large scale bars, but the finer the resolution, the more difficult it becomes to get really great images. So with SEM imaging, real quick, you start with your voltage or your beam source up here. It's going to go through your various lenses and condensers. You're actually landing on your sample right here, and you get scattered electrons that actually form your image that go to a detector, and then you have your image that you see on the computer right there. So as far as sample prep is actually a lot easier with SEM. There's a variety of ways that we can do this. You can either go with tissue or cultured cells and so forth. For cultured cells, one way that we like to do it is that you can actually put a material called ACLAR, which is like a polymer film. You can actually put that down in the bottom of your well plate and grow your cultured cells on the ACLAR in your well plate. Or if you have cells that are in suspension or something like that, we can actually put those onto not a glass slide, but like a little piece of silicon. And the reason why we choose silicon is because it's a semiconductor and it won't trap the electrons that are on there. When you have charging substrates, everything turns bright white and you can't see anything because the electrons can't escape the substrate. So we again do a fix. From there, we let the sample stay in there for a few hours or for 48 hours at 4 degrees C. We do a wash and then dehydrate. And then we put that into our SEM. Now the level of dehydration can vary a little bit. We don't go super dry because you don't want structures to collapse and become, you know, flaky and those kinds of things. But we can't have them like totally liquid. You can't take like a whole cell culture with media and you can't put that into the SEM because these chambers are running under high vacuum, and if you put a liquid in there, it's going to basically sublimate and evaporate and cover the chamber and so forth. So now other methods that we can do, we can also put a thin layer of metal down on top of the sample as well, and that will help with visualizing. But when you do that, you can kind of create some artificial structure. So we try to do as little as possible with these samples as much as we can. Okay, I'm not going to really go over this. The detectors that are on both on the SEM that will give you different types of information. If you're curious about these, I can go into these later on. Okay, here are some examples of SEM images. This is an electrospun scaffold. Oops, hold on. That has actually bacteria growing right here. And this is actually a little spider web here that was made as well. We did, last Halloween, we did a really neat video. We called it Nanoween. It's kind of silly. But anyway, it was looking at structures for Halloween, and we were looking at spiders and creepy things for Halloween. And we put that up on our YouTube channel. So, okay. So if you are interested in doing electron microscopy, but don't have the funds, okay, Kentucky Inbree actually has an avenue to help out with that, okay? There is a call for proposal. Now we are already about to dive into year 2021. Yeah, Martha, help me out here. Is that 2022? Whatever. This year's vouchers are about to kick off in May. The call for proposal actually opens up towards the end of this year. And we do want, it's not just UK, UofL, and WKU. We definitely want people all across the state that are at academic institutions to utilize these vouchers. The voucher is $5,000 from the period of May 1st until April 30th, okay? And there is no extension. When it ends, it ends. But you can really get a lot done for the $5,000. So the other thing that we're having is on August 4th down at UK, we are going to be having an electron microscopy summer workshop as well. This is open to any researchers, even if you're just curious. All of the current Inbree voucher recipients are going to be presenting, and also some past ones as well. We're going to take a tour of UK's EMC Center. We're going to have more in-depth presentations about the capabilities at all three institutions. Tips and tricks on how you can get better imaging, how to correct problematic issues. It's going to be an opportunity for networking and developing collaborations. Martha is going to give, I'm sure, a great Kentucky Inbree program overview. Is that right, Martha?
I don't remember agreeing to that. No, it's fine. Yeah.
I just roped you in. Okay,
Yeah.
and then also how to apply for the 2023 EM vouchers. So with that, that's our information right there. And also, oh, and by the way, the MNTC also has a really big $30 million clean room, which I'm not going to talk about from there. So that's all I got. I'm going to stop sharing. And I'm open for
Great.
questions. You got them too.
Great.
Or no questions, that's fine too.
While everybody thinks of questions, it's 10 microns. The sample thickness should be
Oh, that's
10
good.
Oh,
microns.
10 micron. Okay.
Okay,
For
thank you.
most
That's actually good.
Julia,
Yeah,
did you get a lot of applications this year, sufficient number for the vouchers?
We keep growing every year, and Martha can give more history about this, but in the beginning, it was predominantly from UK and maybe just a tiny few from UofL and maybe none from WKU. This year, we got a lot more from each of the institutions, and we're now to the point where we have to start telling people no. And we've actually, we have more equitable distribution among the 3 institutions as well. So that is a positive growth area.
Any applications outside the 3 institutions?
No, but we would love to see them.
Yes. Oh, they sang SEM, what?
Oh, Dyson, you're muted.
Okay, I got it. Julie, I just sent you a picture of my past work on the
Would it be
Sure.
easier for you just to share that up on your screen?
Yeah, can you just share it on the,
Okay, let me share the screen. Can you see?
Yeah, there we go.
yeah,
Is that the type of image that you can do there?
Absolutely.
So this is a Drosophila, and I saw your spider kind of is that also the image taken at the center?
Oh, yeah.
There? Oh, I see.
Yeah, absolutely. We've also worked with like Norton Children's Hospital, where they had to remove it was a main artery, a main vessel in a child that had a candida bacterial infection. And so when they removed it, it was kind of disgusting, but it was neat to look at under the SEM, because you could see where the biofilm had grown and taken off inside of the vessel. So that was pretty cool. We've been able to see candida cells by themselves. We've looked at red blood cells, all kinds of bugs, cicadas, and cool stuff. So yeah, bring it.
yeah. Can you describe this image, Jason?
Oh, this is some old image that I used during my PhD program, which I'm going to redo this experiment. Wild type is a wild type, and the gene that I was working on, called the DCAP12, is causing apoptosis, which can be partially blocked by inhibitor of apoptosis, suggesting that this gene is somewhere in the canonical pathway. And the point was, this one, I just sent the sample to the core that I was working at, and it was so expensive. And my advisor and I were, oh my gosh. And this is the only image we got because it's so expensive. And as you can see, the numbers are not really great, but again, we didn't have money to redo the images.
So there's some quick question for you. Which institution are you with?
That was a Rochester, University of Rochester.
Yeah, okay. Are you here with UofL now?
Yeah. It was actually several years ago.
Okay.
Yeah.
Yeah, we are, well, here's one thing I want to emphasize is that our facility is, yes, we're set up as a core facility to where we can either do services for you, but we really like to train people and cut them loose, and then they can spend time on the tool and then we don't charge our labor towards it.
Oh,
And
I see.
yeah, I mean, we're an open access facility where you can learn these capabilities, or you or your students. We've taught faculty as well who have an interest in this. They can come and go as they want. It's a 24/7 open access facility.
Oh,
And
I see.
I mean, if you want to come in on Christmas Day and take images of bugs, go for it.
I see. That is basically hourly charge to use the core or the machine.
Yes.
Once you're trained.
Exactly.
I
Now,
see.
yeah, there is, if you have us do it, then there's the cost of the instrument plus also cost of our labor.
That's right. That was why it was so expensive back then.
Right. But if we train you, okay, then it's only the cost of the tool. However, you know, there are training costs. It's the same thing as labor for us. You know, if you use our time, regardless it's for training or services, it's the same rate. But once you get trained, like on the SEM, we can usually train people within 2 hours and cut them loose. On the TEM, it's going to be a little bit less than that. Now, sample prep for TEM is not something that we do lightly. Okay, that's a very, very in-depth process. Okay,
I see.
but once you have the image, once you have the samples ready, we can certainly train you on the TEM, and then you can sit up here for however long you want, and you can use it from there. And also, one other thing I want to emphasize is that if you're in UofL, we have a cap or limit of how many charges or how much your charges can be per month. You can ask Martha about that.
I max it out every month.
Yeah. So what this does is that helps faculty with budgeting their research, so it's not a runaway freight train, because we know that research comes in spurts. You're not on this instrument the same amount of time every month. You know, it'll be like you're getting ready for a journal publication or a poster or a presentation, and you got to get images. So you may be on an instrument for a lot this month and maybe nothing the next month.
I see. All right, excellent information that I need.
Sure. Yeah. More the merrier. Just give us a shout.
All right, thanks.
Yeah, Daesong's in biology, so he could come right over and see you.
Oh yeah. We've got a good number of folks that use our instrument, especially the SEM from biology.
Nice. Good to know.
Okay, well, that was great. Any other questions? This will all be available on the website. All right, thanks.
Thanks,
Thanks,
guys.
everybody.
Thank you.